Good morning. I'm Aurora Savino, a postdoctoral fellow in computational biology in Francesco Iore's group at the Human Technopole. My research project with the Fondazione AIRC is focused on breast cancer. Tumor cells do not grow in isolation. They are part of an intricate system of interactions with other surrounding cells. These cells constitute the tumor microenvironment. The most abundant cells in tumor microenvironment are cancer-associated fibroblasts, which produce the connective tissue sustaining other cells. It has been extensively shown that they can help the tumor grow and metastasize, but they can also help the tumor elude therapies. In my project, I am studying their interactions with tumor cells in breast cancer and how they can impact drug response. The idea is to look at what, in cancer cells, allow them to respond to fibroblast-released signals and to then block these interactions. As a starting point, we are exploiting the huge amount of data that have been collected on in vitro systems, cancer cell lines. We know, for example, to which drugs they are sensitive and which genes they depend on. These data are public, accessible to researchers, and extremely valuable, as they can be reused to answer different biological questions by researchers all over the world. We hope that our research will add a significant piece in understanding the interactions between cancer and fibroblasts, and that it will provide an integrative resource to be exploited by the research community. Indeed, we are acquiring drug response and molecular data about the interaction between tens of breast cancer cell lines, cancer-associated fibroblasts, and several drugs in current clinical use. I believe that making research data freely accessible boosts discovery and should be encouraged indeed. Final goal of this project is the identification of targets and compounds to combine with currently available therapies to prevent microenvironment-induced drug resistance. So we hope to find more effective therapies. I'm curious about how complexity arises from simple components, and one of the main ingredients in creating complexities are interactions. So during my PhD, I have been working on gene networks, as they represent interactions between thousands of genes that can help understanding the status of a biological system. The relevance of this interaction is increasingly appreciated, but its role in breast cancer drug resistance has never been exploited systematically.